Alright, in this second part we're going to talk about combinations of gene pairs with two modes of inheritance, talk about allelic series and lethal alleles. In the previous lecture, part 1, I explained the ABO blood grouping system. Let's now consider the implication of that system on the patterns of inheritance observed. Let's say that two heterozygotes for the IA, IB alleles have offspring. What would we predict or what would we expect in the offspring? Unlike a scenario where we have simple dominance and recessiveness governing the relationship between these two alleles, which we know not to be the case here, in that traditional case you'd have a 3-to-1 ratio of dominant to recessive in the offspring, but we know that that's not going to happen here because we don't have simple dominance and recessiveness, we have a co-dominant relationship. So what we see is a modified pattern of inheritance where we have a 1-to-2-to-1 ratio of both genotypes and phenotypes. Now let's look at an example where you want to predict what could happen in offspring of individuals affected with more than one condition, controlled by more than one mode of inheritance. The mode of inheritance here is codominance. Let's use the example from your text of predicting what the possible offspring might be from when an individual who is heterozygous for both the gene controlling albinism, shown over here, okay, and given the symbolism big A, little a, and the gene that controls the I locus, that glycosyltransferase I mentioned earlier, where we have a heterozygote IA, IB, shown over here. Okay, so this portion of the figure shows what to expect if you're just tracking inheritance of either the gene controlling albinism or the gene controlling ABO blood typing alone. Now let's consider what would happen if two individuals who have these genotypes shown up here in the top were to have children. So they're each the same with regard to genotype. They're heterozygous at both loci, both the A loci and the I loci. And when it comes to the I, their genotype is IA, IB. So the easiest way to think about this is to consider it one gene at a time and use a fork diagram. And that's what this figure depicts. So in this left-hand portion of the fork diagram, we're first considering transmission patterns in the A gene that controls albinism. That is a very straightforward dominant to recessive relationship between big A and little a. So we know that if you have heterozygotes and they cross or mate, that you should expect or be able to predict that their offspring will show a 3-to-1 phenotype ratio, 3 parts pigmented or colored normally to 1 part albino or non-pigmented. So 3/4 to 1/4, and so that's what the authors have broken out here in making this figure. Then we move on to the next part of the fork diagram here. This column addresses the transmission patterns expected with regards to the I locus or gene. So in that case, we know that we have a modified pattern. Okay, and we know that it is 1-to-2-to-1 or 1/4 type A, 2/4 or half type AB, 1/4 type B. Those are the probabilities of those types of offspring. So we simply lay those into this forked diagram in two positions, here and here, okay? And then we come up with the final column, which is the probability of various offspring. So in the final column, we're asking what's the probability at the top of being pigmented and type A, or pigmented and type AB, or pigmented and type B, and so on, okay? And we get these probabilities, here 3/16, by multiplying 3/4 by 1/4. 3/4 by 1/4 equals 3/16, okay? And if you do that and you add up all of the numbers in this column, you should get 16 out of 16, or a perfect probability accounting for all possible offspring type. Now look at the phenotype ratios shown down here. They are pretty complex, essentially 3 to 6 to 3 to 1 to 2 to 1. That's a lot more complicated than the 9 to 3 to 3 to 1 ratio that you would expect in a scenario where you were tracking two genes that is sorted independently, and you were tracking two alleles, and one was clearly dominant to the other. Much more complex pattern of inheritance. Many genes have multiple alleles, and this is usually or often referred to as having an allelic series. The relationship between the alleles can be somewhat complex, needs to be thought about one part at a time. It's very important to note that you have to consider the relationship between alleles one pair at a time. For example, just because a wild-type allele is dominant to one other allele doesn't mean it is dominant to all other alleles for the same locus or gene. You have to think of them and study them one pair at a time. Your text gives you the example of the white gene in Drosophila, which controls eye color. I'd like to reconsider the gene that encodes that particular gene, which, as you know, I told you before, encodes the enzyme tyrosinase, which we already discussed when we talked about albinism. Albinism is associated with a severe loss of function mutation, and the allele that's associated with that, it's a classic example of a loss of function or a null allele, and it is typically expressed using the lowercase c alone, shown here on the right. So that rabbit over here on the right is showing the classical albino phenotype, pink eyes, white, completely white fur everywhere, a complete lack of ability to make melanin because the tyrosinase encoded by the little c allele is totally non-functional. So there are other alleles for this particular gene besides the little c shown on the right and the big C shown over here on the left, or wild type. So those other alleles include the CCH, or chinchilla allele, and the CH, and those are upper superscripts, which is also called the Himalayan allele. And so each of these is encoding tyrosinase. They're all forms of the same gene, and they have been very well studied. So we know that in the wild type, or the big C, we have fully functional tyrosinase. So it is able to produce melanin normally, and we see normal pigmentation, and it is fully dominant to all of the other alleles shown in this scenario. The lowercase c, as I discussed before, is completely inactive tyrosinase. It's a fully recessive allele. So when you are homozygous, as this individual is here, for that lower c allele, you are albino. You don't make any normal tyrosinase, so you can't use it to make any melanin whatsoever. The chinchilla and the Himalayan are a little bit more interesting, at least in my opinion. So the chinchilla, or the CCH allele, it encodes a partially active tyrosinase. So you've got tyrosinase, and it works a little bit, and so you can make a little bit of melanin and have a little bit of pigmenting. And so the result of that in the homozygote, shown here, is a somewhat grayish rabbit. And by the way, these genes occur in other mammals as well, so they occur in rats and a variety of other animals, and they show similar patterns in coat coloring. But you see a rabbit that's got a little bit of pigment in it, and it's got actually normally pigmented eyes. And this will be observed in individuals that are homozygous, such as this one, or individuals that are superscript CH and lowercase c, which tells us that the chinchilla allele is dominant to the albino allele, the lowercase c allele. The Himalayan allele, CH, shown over here, is really interesting. It produces a temperature-sensitive tyrosinase, such that that tyrosinase is only active in the cooler areas of the body. So a rabbit's ears, nose area, and little paws are typically, they're the extremities, and so they are cooler than the other parts of the body. So the tyrosinase that is produced there when genes are expressed in those cells is functional, so you get pigmentation there. In the hotter parts of the body, including the eye region, the tyrosinase is non-functional, and so you're left with no pigment there. And so the overall pattern is referred to as Himalayan. So it's a temperature-sensitive allele. Very interesting. Individual studies where crosses between these rabbits or other animals showing these alleles are done where allowing such that you're tracking just 2 alleles at a time have shown that the relationship between the alleles is what I have depicted here. The wild type or uppercase C is fully dominant to all other alleles. The lowercase c alone associated with albinism is fully recessive, and the relationship between the chinchilla and the Himalayan alleles is shown here. So you can think of the chinchilla allele as being able to trump or be dominant to the Himalayan or the albino allele, and you can think of the Himalayan allele as being dominant to the albino allele, but recessive to the chinchilla and the wild type uppercase C allele. So it's a somewhat complex story, but if you break it down one allele pair at a time, and you understand the molecular basis underlying the story, it becomes a little bit easier to follow. This figure depicts the genetic crosses that were done in order to establish the relationships between the C alleles. Note that they're tracking one allele pair at a time using traditional Mendelian crosses. And note that in the F2, we're seeing that in all cases we have a 3-to-1 traditional ratio that establishes which phenotype and thus which allele is dominant to the others. Let's now consider lethal mutations. Lethal mutations result in lethal alleles. And these are mutant alleles that are usually but not always recessive, and they are alleles where in the homozygote they cause fatality. And they'll cause fatality at the time of life that wherein the gene product is necessary. So sometimes they cause fatality at the embryonic or fetal stages, sometimes later on in life. But the point is that at some point you need that gene product and it's not there, and so lethality or death occurs. Lethal mutations are easily observed in haploid stages when you're doing things like plant genetics, where you can actually study the gametophyte stage or the haploid stage. They're also often identified when you are doing crosses and you notice that an entire phenotype class that you predicted would occur is missing or absent. And we'll look at an example of that now. The Agouti gene is a gene that controls patterns in coat coloring, and it's been very well studied in mice, although it does actually exist in other animals. The Agouti phenotype is shown in the top part of this figure. So in sort of normal or wild type mice, we see this banding pattern where pigment's been laid down in different portions of the hair shaft making up the animal's coat. The Agouti phenotype is the sort of normal or wild type phenotype, and when you have mutants, a particular type of mutant in the Agouti gene, it has been observed as having uniform yellow hair color, so there's no banding pattern in these hair shafts shown here. Geneticists noticed when they were first studying this that if you cross Agouti by Agouti mice, the progeny are all Agouti. Okay. And if you cross yellow by yellow mice, the progeny are 2/3 yellow and 1/3 Agouti. And if you cross Agouti by yellow, you get half yellow and half Agouti. And these are not typical Mendelian ratios. So after doing these crosses for a while, they proposed the hypothesis or idea of a lethal allele, the idea that whatever allele was controlling yellow was also resulting in lethality or death. So they proposed that the Agouti, the mutant allele, which they assign the symbol A superscript Y, shown over here, okay, is in fact a dominant mutant allele that results in yellow mice rather than agouti-colored mice, and they proposed that homozygotes for that allele could not survive, hence the term lethal allele. So they explained their results in the manner shown in this figure. If we start out with the right hand or B part of the figure, if you look at the yellow by yellow cross, okay, they proposed that the genotype for those yellow mice was actually heterozygous, shown here, and that when you cross them, these are the possible offspring that result. The agoutis are big A, the yellows are the heterozygotes, shown here and here, and that anything that was homozygous for the mutant allele actually died, and so it wasn't observable as a phenotype class, and that would then explain the 1/3 agouti, 2/3 yellow mice resulting from this cross. And then they did additional experiments to support their hypothesis. So when they hypothesized that when you were crossing agouti by yellow, that the agouti mice were usually big A, and that the yellow were big A, they were the heterozygous, big A and A superscript Y, as shown here. If they reproduce, then the results of the crosses are shown here in this Punnett square on the left. The agoutis are all homozygous for the wild-type allele, and the yellows are heterozygous for wild-type and the mutant yellow allele. And this fit with their 1/2 to 1/2 agouti to yellow pattern observed for those particular crosses, supporting their hypothesis. The structure of the agouti gene and the mutation event that gave rise to the A superscript Y mutant lethal allele is now well understood. So the gene structure associated with the agouti gene in the wild type or with the A allele is shown up here on the top. Okay, there's the agouti gene, and it controls the patterning and hair coloring seen on the hair shaft. And it is controlled, its expression is controlled by a promoter. You should know what a promoter is from your previous biology classes. Okay, the agouti gene is normally expressed in a manner that is regulated. So it's normally regulated, so that this promoter actually is controlling, when and how often and how much the agouti gene is used to make the agouti gene product. Now, just upstream of the agouti gene is another gene called the rally gene. It produces a protein necessary for normal mouse embryonic development, and the rally promoter controls the expression of the rally gene. And the rally gene is, the regulation of the rally gene is different than the regulation of the agouti gene, and the pattern of regulation is somewhat complex, and it involves the rally gene product itself. Okay, but the point here is that that's the normal structure, and in the normal scenario with the A allele, we have regulation of gene expression so that the agouti gene is not turned on full stop all of the time. Now, in contrast, down here, we have the mutant allele, and that mutant allele is an example of a deletion mutation. So we lost about 120,000 base pairs by deletion, giving rise to an altered gene structure. We still have all of the agouti gene, but we've lost the agouti gene promoter, and we've lost the rally gene. So now the rally promoter is actually hooked up to the agouti gene. And the problem here is that in this scenario, that agouti gene is expressed constitutively. Or all the time. Full, constant expression. And that is a problem because in organisms where that is happening, there's far too much agouti gene product. The result is lethality in the embryonic stage in the homozygote, or very altered mice as observed by the yellow phenotype in the heterozygote. The agouti gene is a really fascinating gene, and there have been a lot of studies done looking at it. It's an example of a lethal allele. or it's an example of a gene that has a lethal allele, but it's also an example of something called pleiotrophy, or a pleiotrophic gene. And that means that alteration of multiple phenotypes can occur when you have a mutation to that single gene. The Agouti gene is the classic example of this, and in part, here's why. Those yellow mice are different not just in their coat coloring, but they're also showing differences in other biological tendencies or phenotypes. For example, yellow mice are highly prone to obesity, and this picture here shows a very obese yellow mouse that's been fed exactly the same diet and exercised the same amount as this wild-type mouse. And it turns out that they have other health problems too. They're very prone to diabetes, and they're very prone to cancers. We know now that the Agouti gene affects methylation patterns in the mouse genome, which controls expression of a variety of other genes, therefore affecting phenotype in many complex ways. So we refer to this scenario as pleiotrophy. Here are some possible study questions for you to consider fair game on a quiz or an exam. One, you should be able to solve inheritance problems involving two genes that show different modes of inheritance. So one mode might be traditional Mendelian inheritance, another might be the mode of inheritance shown in codominance stories or scenarios. Two, you should be able to define the term allelic series with an example, or two, if you're studying your textbook. Three, you should be able to understand the molecular basis for the tyrosinase allelic series, the example that I gave you with the rabbits involving the chinchilla and the Himalayan alleles. You should know what the gene product is, it's tyrosinase, and you should know what the four different alleles encode with regards to tyrosinase and how they affect phenotype. 4, you should be able to define the term lethal allele with an example. We discussed the Agouti allele in this past lecture, and it's in your textbook. 5, you should know the difference in gene structure in the Agouti A versus A superscript Y allele, and what effect that gene structure has on phenotype. 6, you should be able to define a pleiotrophic gene with an example. And that concludes this part of the lecture.